Namaskar, welcome to PK's classes. Today we are going to study about the mechanism of resistance to aminoglycoside antibiotics. So before discussing the mechanism of resistance, let us first discuss the mechanism of action. So to know about the aminoglycoside antibiotics and its mechanism of action, you can refer to our earlier classes. The mechanism of action says that aminoglycoside antibiotics bind to the A site on the 16S ribosomal RNA of 30 subunit of prokaryotes or bacteria to this site. So this is the A site in the 50th subunit where the codon-anticodon pairing occurs, codon of messenger RNA and anticodon of tRNA pairing occurs here. So initiation of the protein synthesis occurs. So aminoglycosides, by inhibiting the A site on the 16S ribosomal RNA of 30 subunit of prokaryotes, inhibit initiation of protein synthesis, and that results in incorrect codon-anticodon pairing, so inhibits protein synthesis. So if we will discuss the mechanism of resistance of aminoglycoside antibiotics, there can be four major mechanisms. In addition, there can be some other mechanisms also. Major mechanisms is the modification of ribosomal target, just discussed, the 16S ribosomal RNA of 30 subunit of ribosome. Second is the modification of the aminoglycoside antibiotic itself. Third is reduced uptake, so there will be decreased influx. And fourth is increased efflux. So the bacteria will throw out the aminoglycoside antibiotic. So throw the antibiotic out of the bacterial cell. So let us discuss one by one. So the first mechanism is the target site modification or ribosome modification, or the 16S ribosomal RNA methylation. So methylation of the 16S ribosomal RNA occurs. Methylation of the 16S ribosomal RNA occurs, and because of that there will be no binding of aminoglycoside antibiotics with the target. So the enzyme here required is 16S ribosomal RNA methyl transferase. So in short, 16S ribosomal RNA methyl transferase, in short RMTH. So this enzyme methylates nucleic bases at the A site on the 16S ribosomal RNA of 30 subunit of ribosome. So there will be no aminoglycoside binding with the target. This leads to resistance. So this RMTH is encoded by genes like RMT, which may be RMTA, RMTB1, B2, C, D, E, F, G, H. So there are two classes of RMTH, those that affect 4,6 disubstituted aminoglycosides and those that affect 4,6 and 4,5 disubstituted aminoglycosides. So for that you can refer to our earlier classes on the classification of the aminoglycoside antibiotics. And the structure wise we can have four classes of aminoglycoside antibiotics: those without 2-deoxystreptamine ring, and this is the structure of 2-deoxystreptamine, and there can be monosubstituted and 4,5 disubstituted and 4,6 disubstituted. So in this case those RMTases which affect 4,6-disubstituted aminoglycosides via methylation of the N7 position of nucleotide G, and those that affect 4,6 as well as 4,5-disubstituted aminoglycosides through methylation of the N1 position of the nucleotide A. So in this mechanism, the molecules which are affected belong to these two categories, 4,6 and 4,5-disubstituted. Then the second mechanism is modification of aminoglycosides by the aminoglycoside modifying enzymes, or AMEs. So three types of enzymes are there: N-acetyl transferase, aminoglycoside N-acetyl transferase, O-phosphotransferase, aminoglycoside O-phosphotransferase, and aminoglycoside O-nucleotidyl transferase, mainly adenylation occurs. So acetylation by acetyl transferase, phosphorylation by phosphotransferase, and adenylation by adenyl transferase or nucleotidyl transferase. So they cause the acetylation of the amino group and phosphorylation and adenylation of the hydroxy groups at various positions in aminoglycoside scaffold, which decrease the binding affinity of the drug for its target, and that causes the loss of antibacterial property and hence resistance. Then we will go to the third point. The third point is the change in membrane permeability, and change in membrane permeability causes the failure of penetration of the aminoglycoside antibiotic into the bacterial cell, and for that we must know how aminoglycosides permeate, and you can see these porine-like structures. They are responsible for entry of the aminoglycosides into the cell, and one is porine channels, this one, and the other is simple diffusion through the phospholipid bilayer because of the electrostatic binding, because the aminoglycosides are cationic, whereas the bacterial membrane is negatively charged in gram positive because of the presence of phospholipids and teichoic acid, and in gram negative bacteria the phospholipids and lipopolysaccharides, and they make the bacterial membrane negatively charged. Aminoglycosides are positively charged, and because of that there will be electrostatic binding and simple diffusion occurs. So two mechanisms by which aminoglycosides permeate into the cells. One is the porine channel, second is this one. So how resistance occurs? Resistance occurs due to structural changes in the porins and changes in composition of cell membrane lipopolysaccharides. So change in lipopolysaccharides, so that means affected gram negative bacteria, and porins structure change also causes the resistance. Then the fourth mechanism is active transport or efflux pump. So there is resistance nodulation division or class of efflux proteins, R&D family of efflux proteins or efflux pumps. For example, ACR-AD, MEX XY-OPR-M. So ACR-AD in gram-negative bacteria and MEX XY-OPR-M in Pseudomonas aeruginosa. These are some of the examples of efflux pumps. So besides these four mechanisms, there can be other mechanisms, gene mutations, and some of the genes which are responsible for resistance because of mutation. Streptomycin, 3 genes have been identified: RRS, RPSL, and GIDB. RRS is also responsible in amikacin and kanamycin resistance. So mutation in these genes can cause resistance. Then the sixth one is the membrane proteases. As we have discussed in the mechanism of action, aminoglycosides cause misreading of the mRNA, so there will be membrane disruptive proteins are formed, and because of the membrane disruptive proteins that promotes the action of the aminoglycosides. So bacteria like Pseudomonas aeruginosa, which shows resistance by this mechanism, they develop proteases which cause the proteolysis of these membrane disruptive proteins, and this promoting action of aminoglycosides are inhibited. So that leads to resistance. And then the seventh mechanism is the biofilm formation, which is causing the adaptive resistance. So the bacteria develops a biofilm which prevents from the action of the antibiotics. So suppose the aminoglycoside antibiotics, they now cannot act here. So this biofilm protects the bacteria from the action of any harsh environment, including the action of antibiotics. So this is also another mechanism of resistance. So this is all about the mechanism of aminoglycoside resistance. Thank you.